In our previous video, we have understand that mature GPI-APs are expressed on the plasma membrane, both in the case of wild type and also in the case of this dual knockout, it is Calnexin and calreticulum dual knockout. However, one more thing that we have observed in the Calnexin and calreticulum dual knockout results is that there are some immature GPI-APs are also expressed, which are not being cleaved off by PI-PLCs. So in this video or in this experimental results, we will understand that whether these GPI-APs, non-glycosylated GPI-APs also use this Calnexin dependent mechanism for deacylation, or there is some independent mechanism. So in general, this more than 95% of the GPI-APs has at least one N-glycosylated site. So in this case, what they have done, they did is they used two different construct where they have modified the N-glycosylated site, and first one is modified with LY60 with a his tag, and second one is modified with this GFP GPI. So both of them could not be glycosylated. So when they express this construct in the wild type cells, they can see that it can be cleaved off by PI-PLC. That indicates that they are not been that indicate that they are not dependent on Calnexin, as since it does not have any N-glycosylated site, it will not be able to interact with the Calnexin. So there is some non-Calnexin independent mechanism for deacylations, and they observed this thing both in the both construct, that is in the his LY60 and his GFP GPI. And the surprising thing that they observe is that similar results are shown for the Calnexin and calreticulum dual knockout. That is, there is almost negligible sign for immature GPI-APs expressed on the plasma membrane of the dual knockout. And these things are being validated using the quantifications of this flow cytometry results, that the difference between remaining GPI-APs after this PI-PLC cleaved off is non-significant in the case of wild type, and also in the case of this dual knockout. And these things are observed for this LY60 and GFP GPI. So after validating this, that this his LY60 and his GFP GPIs has this Calnexin independent mechanism. They also checked it for the CD59. And for CD59, what they have find out when they modified the N-glycosylated site, that is N43Q, what they have find out for the compared to the wild type, they see some increase in this remaining PI-PLC, or we can say that there are some increase of this immature GPI-APs expressed on the cell surface compared to the wild type, both in the case of this CD59 wild type and also in this CD59 and 43Q. And that can be observed when we quantify this panel B flow cytometry results. We can see that in the wild type CD59 there is a expression, there is a high expression of this remaining GPI-APs. This indicates that there are more immature GPI-APs are expressed in the case of this CD59 wild type. So this could be because it requires Calnexin for deacylations. However, this difference is significantly reduced when the N-glycosylation sites are being modified, where the statistical significant difference is reduced by has a probability of less than 0.03, whereas in the case of the wild type CD59 there is a probability, the false positive probability is less than 0.01, and this indicates that there could be some independent mechanism or Calnexin independent mechanism for deacylations for the non-glycosylated GPI-APs. So from there they made from these experimental results, they made a point that there could be some independent Calnexin independent mechanism for deacylations for the non-glycosylated GPI. So I hope you understood that how they have come to this point, the conclusion from these experimental results. And if you need to understand more about this, these experimental results in detail, or the methods that they have used for this experiments, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.